Hey, good evening, everyone. This is the 40th annual meeting of the Hudson Hoagland Society. Wow, I just have to think back to 1985 when I became president of the Worcester Foundation. My development officer walked into my office and said, "Are you going to keep me?" I said, "Well, why wouldn't I? I don't know anything about fundraising." And he created the idea for the Hudson Hoagland Society, and our first meeting was in 1986. Welcome you all. This is a very pressing time in our profession, and at no time have we ever felt more indebted to you and your generosity for the support of the basic science that underlies so many medical advances. I don't want to go into a lot of detail. I just want to tell you that the senior leadership, Chancellor Collins, Dean Flott, and many others here have been working night and day to be sure that your investment in us is matched by federal investment, which is under attack. I don't want to give a political speech, but just wanted to emphasize how uniquely important your contributions have been this year. I'd like to not only welcome you, but show you the four recipients of Worcester Foundation grants this year. And I think here they are. We had 39 proposals. I'll say more about that in a moment. Dr. Hemming is working on the other kind of gastrointestinal cancer. This is not the major, when you hear somebody has stomach cancer. This is a major form, but not the most major form. He's got a whole new idea for targeting the molecular deficiency in cell regulation that gives rise to these particular so-called stromal tumors. Dr. Watson, I presume, she's a brilliant young investigator that we hired a few years ago, and she's got a new angle on some rare cancers. Now, the rare cancers, you might say, well, how rare are they? And your empathy is stretched a little bit, but of course, as humans and as physicians and scientists, we should never be stretched, because if you have a loved one or yourself have cancer, it doesn't matter how rare it is. She has got some very promising new ideas about how the genes that cause these cells to go in the wrong direction arise because genes that can cause cancer by themselves come together and fuse, and thus make a worse storm, a less perfect storm, or you could say, if you're wanting cancer, a more perfect storm. Ching Yu, he is the recipient of the Bassick Family Foundation Award, and I want to thank Jack Bassick and his family for their continuing support of making this special grant possible. His title shows the attention to grammatical detail that a gifted scientist can have, but I'm going to put it in simple terms for you. Inside our cells, we've got proteins of various kinds. Most of them do not do their job all alone. They're not only children like I am. They collaborate with a nest of other proteins with which they interact. And it is that interacted structure of 5 or 6 proteins that is the functional entity. We've known this for a long time. You might say, what's new? We knew it because we would pull these things out of cells and study them. But the problem was that sometimes they would glop things together that weren't really there in the real living condition. And what Ching Yu has done is developed a platform of technology that allows him to go into living cells, both single cells and tissue, to see what proteins are really interacting with other proteins. This is very promising. It is called a protein interactome, but the key point of this project is that it's being done in living cells. And then Jingzhan. Well, guess what? People that use artificial sweeteners. I didn't know this, and I love reading these proposals. Um, they 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 do well because there's less caloric intake. However, many of these people have a sweetness addiction, not a sugar addiction. And so they eat pastries and cakes that may have artificial sweeteners as the sugar, as the sweetener, but also have fat in the frosting and other caloric commodities, and they're wolfing it in. Sweetness addiction is not a sugar addiction. It is due to a particular affliction of the tongue. Now, again, I learned so much from reading these. What Jin Zhang has discovered is that when people have this addiction to sweetness, their tongue cells become repopulated. There are about 30 different kinds of cells in the tongue. And these people have the sweet sensing cells become overpopulated in the tongue. This is a radical idea. And it's something that I think has tremendous clinical and societal nutritional impact. So those are the grantees this year. The other 35 were equally outstanding as science, but the committee of one, had to make judgment calls on exactly why these are the most transformative in principle. I corresponded with every one of the other people and told them in detail why their proposal was great and why I'm going to try, as will their chairs and other colleagues, support their programs going forward. Finally, I do not usually say much about the speaker. We leave that to the introducer, which in many years is Dean Terry Flott, who is with us tonight. Terry has been, as I said, leading with Dr. Collins our efforts with the attorneys generals and all the other things we need to do to marshal our efforts to support basic biomedical research at this time. Dr. Collins, in a moment, will join in welcoming you with me and will introduce our speaker. What I wanted to say is something that just occurred to me this morning. It is exactly 60 years ago this month that as a graduate student, I wrote in a lab notebook my first experiment on RNA. And that has been my career focus in research for these 60 years. Had an angel come down to me that day and said, Thoru, you will spend the next 60 years working on RNA, but what you don't know is the last 3 decades of that research, you will be spending it at the greatest citadel of RNA biology in the United States, and you will have colleagues, the likes of Victor Ambros, a person whose science is beyond description, although the chancellor will describe it and Victor will describe it. But it was a person of human character that endears me greatly to him. So forgive me, Dr. Collins, for preempting just a wee bit of your introduction. And now I would like to turn it over to Chancellor Collins, who is the chancellor of our medical school. He's also professor of population and quantitative health sciences in our medical school and serves as well as senior vice president for all the health sciences for the entire University of Massachusetts. And I'd like him now to offer his welcoming remarks and then introduce our speaker. Chancellor Collins.
Yeah, thank you very much, Dr. Peterson, and good evening, everyone. Let me thank you, Thoru, for 40 years of leadership, really quite extraordinary, and to our colleagues in the Office of Advancement for once again bringing together the Hudson Hoagland Society. It's very heartening today to be able to join today's virtual event and to be among the stalwart supporters of biomedical research. I think all of us can recognize that despite the challenging and uncertain funding environment that we find ourselves in today, we really must manage through these challenges, continue to benefit from and be inspired by the incredible support and generosity of our benefactors, exemplified most recently by leading philanthropists like the Damari family, based in Florida, who this year made a $35 million gift commitment to UMass Chan to support cutting-edge neurodegenerative research that's taking place on our campus, and to name our newest research facility the Paul J. Damari Center. This type of support and partnership by so many of you is more essential than ever, and really critical to us as a result of the disruptive changes we're now experiencing in federal support for biomedical research, and we're not exactly sure what the future lies for funding from the federal government. Now, more than ever, it's important for us to seek out partnerships with and support from those individuals, organizations, and foundations that understand research's value proposition, those all of whom can help us to continue along our growth trajectory. We're fortunate indeed to have a growing cadre of supporters who choose to invest in UMass Chan, and more specifically, our world-class and Nobel Prize winning faculty. Our fiscal year ending June 30th will be one of our most successful fundraising years in our history, with many new endowed scholarships and endowed chairs being established to support our promising learners and our preeminent faculty. In a time where federal funding is at risk, the fact that so many individuals and foundations are willing to support the medical school is indeed most encouraging. And we look forward to marking the culmination next week of another really wonderful academic year by coming together with our commencement festivities on June 1st. Our newly minted physicians, nurse practitioners, and scientists are poised to make positive and lasting changes in their chosen professions. And I have to be honest with you, we are very proud to call them one of our own. So too will we be proud and privileged to bestow honorary degrees on Marcia McNutt, PhD. Dr. McNutt is the president of the National Academy of Sciences. One of our alums, Carolyn Clancy of the class of '79, the Assistant Undersecretary for Health in the United States Veterans Health Administration, who was instrumental in assisting us to be able to build and now serve our Central Massachusetts residents on our campus. And Majid Jafar and Lynn Bargu Jafar, co-founders of the Lulu Foundation, a foundation which supports research in a rare disease, a single gene defect disease, CDKL5 deficiency. Truly, we have built something very special and spectacular at UMass Chan. We believe we are an academic and research environment worth protecting and worthy of investment. It's very special because our collegial, collaborative, passionate, and renowned colleagues like our own Victor Ambros help shape and sustain an amazingly dynamic, fertile, and caring culture here at UMass Chan. This evening, you'll have the tremendous privilege of hearing Dr. Ambros speak, and now I have the wonderful privilege of introducing him. Dr. Victor Ambros is the Silverman Chair in Natural Sciences and Professor of Molecular Medicine at UMass Chan. Together with his wife, Rosalind, Candy Lee, as we know her, they joined UMass Chan and our burgeoning RNA community back in 2007, following stints at Harvard Medical School and Dartmouth College. Dr. Ambros was one of our first big recruits, if you will. Following stints at Harvard Medical School and Dartmouth College, we were very fortunate, and I think a bit lucky, to encourage Dr. Ambros to come and join our community. In fact, I remember in one of the final visits we had, I said, Dr. Ambros, you could go anywhere in the world. Why UMass? And he said very simply, I think we owe it. We think we owe it to science to bring together all the great RNA biologists together to create an RNA Therapeutics Institute, and we were so thrilled that he accepted our offer to join UMass Chan. Dr. Ambros' innate curiosity, ingenuity, and persistence have forever changed the world's understanding of RNA biology and gene regulation. His lab's discovery of microRNA and an evolutionary process and fundamental biologic mechanism that regulates gene activity in all living cells was first published in a seminal 1993 Cell paper. His trailblazing work in elucidating the role of microRNA garnered him science's most prestigious prizes, including a Lasker Prize and a Breakthrough Prize. On October 7th of 2024, past October, the Nobel Committee at the Karolinska Institute in Sweden placed an exclamation point on a truly stellar research career by selecting him and his colleague, Gary Ruvkin, as co-recipients of the 2024 Nobel Prize in Medicine or Physiology. I have to be honest with you, the last several months have been a whirlwind for Dr. Ambros and his family, but we'll be forever grateful to him for shining a light on UMass Chan as he has done on so many different occasions and raising the profile of our institution across the globe. It's because of faculty members like Dr. Ambros that UMass Chan has become such a globally respected research powerhouse. This evening, Dr. Ambros will be delivering a talk entitled Exploring the RNA Frontier and Why It Matters to You. And I hope all of you now will join me in welcoming Dr. Victor Ambros, this year's Hudson Hoagland speaker. Victor?
Thank you. Thank you, Chancellor Collins and Dr. Peterson, and for these introductions. And thank you, Kim and the Society, for the invitation and opportunity to speak with you today. I'm going to share my screen and see if that works. So I can hope you can see that. I want to get rid of this little thing there. Tell me if it doesn't look good. So
it looks good.
Great. So I'm Victor Ambros. I'm here today with Rosalind Lee, Candy Lee, who's my wife and longtime partner and collaborator. And it's our pleasure, to be here with you today. I want to talk a little bit about who I am, right? And so it was the first slide showed where I came from. Let me see. There we go. I think things are going to be a little slow, maybe. But okay, this is me. I started out growing up in Vermont, and I started out as a scientist. I can't remember when I, wasn't a scientist in my aspirations. And so I went to college, and I went to MIT, and this was a place where I was influenced surprisingly by other students who converted me from an aspiring astronomer to a molecular biologist. And I had really some wonderful experiences working in laboratories, doing original research. And this guy, Ed Gruberg, was a postdoc. And working with him, I published my first actual paper. Then I did postdoctoral, a PhD work again at MIT with David Baltimore. There I studied poliovirus. And then I went on to study postdoctoral research again at MIT, and this time with Bob Horvitz. And that's where I started working on the animal that I'm working on today as a model organism for the studies of developmental biology that I'm doing. And so then I got a professorship at, I became faculty at Harvard. Was there a while. Then we moved to Hanover, to Dartmouth. And then finally in 2008, as the chancellor described, we moved to UMass. And I'm here to tell you that UMass is the best institution I've been at in a lot of ways, and most prominently because of the ease with which we at UMass share ideas, and inspire each other and support each other and collaborate. So it's fun to do science at UMass, and UMass does great science in large part because of the culture and the community that's been developed here. And the community here is great because of the leadership of that's here now and who has been here for the last 40 years or so. So I run a research lab that studies biological questions that interest me. And that's one of the things I want to emphasize here is sort of, let's remind ourselves how science works, right? And so the main unit is the laboratory at a university. And the laboratory is led by a principal investigator. That principal investigator has been hired, in my case, by UMass. And UMass, and the lab is supported by federal agencies, by the NIH, NSF, and others, right? So UMass provides infrastructure, and this comes from funding from the American taxpayers and philanthropy. And the federal funding, of course, is a little bit more in dollars, let's say, than what comes from the university. That originates with the American taxpayers, of course. Importantly, I don't know if you can see my pointer, hopefully you can. This funding originates in the mind of the investigator. The investigator has ideas about matters that seem important to that person and that they're very curious about, okay? So the principal investigator brings a certain perspective on science to the university and a certain unique set of interests and formulates projects that are then vetted by peers at these federal agencies, and that's where the funding comes from. Now, who in the laboratory actually does the work? These are largely trainees and students, PhD students primarily, some undergraduates, even some high school students in some labs, postdoctoral fellows, professional scientists. All of these people are doing the work and contributing to the productivity of the lab in terms of publications, patents, and so forth. And what that is communicated to this public is essentially new knowledge of biological mechanisms. So that's the output of the lab. And that has consequences that pay off in terms of deeper understanding of disease, and therefore novel diagnostics and therapeutics can result. Okay. Now, the results or the productivity of the lab is what feeds back and reinforces the funding from the federal agencies. Okay. And so this cycle of re-proposing and revising and evolving ideas coming from the lab are a product of the work of the trainees in conjunction with the investigator, shaping the science that they do and producing results which then are judged by their peers to be substantial and impactful, and therefore reinforcing that funding cycle. So what are the societal benefits of this? Let me see. How do I get rid of this little guy? No. I bet I can move this thing. All right, let's put it. Is there a way to get rid of it? Let me see. Is there
just move it down.
Okay, I just move it down. Societal benefits, right? And these include, obviously, from the new knowledge and novel diagnostics and so forth that can stem from the research in the lab. Also, it's not often recognized that an economic stimulus comes from this overall activity. It may not be from my lab, but in aggregate, all the labs, let's say at UMass and elsewhere, there's an economic stimulus that balances and supplements what was put in. And so it's been calculated that about 2.5 times what's funded from the federal agencies comes out from this research into society. Now, also what comes out is people. And these trainees then go on to academic and other positions in law, journalism, public policy, pharma, and so forth, management in pharma, research in pharma. And so the trained scientific workforce is another societal benefit. Now, I thought it'd be, interesting to kind of consider this. We all sort of know about this, but maybe we don't always reflect on it carefully. So this is a U.S. scientific research ecosystem. And the key of this whole process is ideas that come from individual people's minds. A lot of this is, what are we curious about, right? And so in our lab, we're curious about how animals get their shapes. We know that the DNA of these animals confers a developmental program that's unique to the animal, and that results in a unique shape, okay? And the mystery here is, how does the DNA contain the information to make, with such perfection, a pig or a rabbit or a person or an insect or a nematode? And the nematode, C. elegans, is the model that we use in our lab to try to get at these questions of how the DNA specifies an animal. So this is the model organism idea. And it's based on the idea that, about observation, that from all genome sequences, we know that animals have about 20,000 genes, right? Humans do. And all these other animals do as well, by the way. And they all share about 10,000 genes. So in other words, the common ancestor of all these bilaterian animals, some 500 million years ago, had in its genome a genetic toolkit, much of which is intact and functioning in the genomes of all the animals today. So the idea then is that the molecular machinery inside animals, the genes and gene products, is largely conserved, and evolutionary diversity reflects diversity in how the machinery is regulated. So by studying these model organisms, we will study at the same time what's in common amongst animals, let's say what's in common between C. elegans or a fly and humans, but also we can understand better how it is that animals have such astonishing diversity. So that's the model organism idea. And for us, the worm is our model organism. And so the idea is that if we study genes of the nematode, we will learn about human biology. And so here's our model organism, C. elegans. The big ones are the adults. These oval things are embryos that one of these adults has deposited in this milieu here, which is the bacteria that they eat. And you can see larvae, embryos in the eggshells, and then larvae of various sizes. It only takes about 2.5 days for an animal to develop from an embryo to an adult. So it's a very rapid life cycle, and that facilitates experiments. We can carry out 2 genetic experiments a week, for example. Now, this is the video that really got me hooked on C. elegans. Whoops. Yeah, let's go back to the video. 1 cell, 2 cells, 4 cells. This is sped up. This process is taking about 16 hours in real life, but notice how that first cell is now about 120 cells. Maybe now it's about 240. And by the time, right about now, all the cells are made, there's 550 cells, and they start to rearrange themselves into a worm, right? And if you just reflect on that for a moment, that's pretty amazing, because all the information in that, it was in that egg to make that worm, right? 1 cell, 550 cells, and they all have identical DNA. So what's different is what's happening is that different genes go on versus off. So in a nerve cell, nerve genes are on, muscle genes are off, and intestinal genes are off. In a muscle genes are on, nerve genes are off, intestinal genes are off, and in the intestinal genes are off, specifically on. So basically, this story of the development of an animal, we've come to understand, is a story of regulated gene expression. And so how does that work? So we want to understand how regulated gene expression works. We need to reflect a little bit about individual genes. Now, what does a gene do? The gene is a piece of DNA that encodes for an mRNA, a messenger RNA. And that messenger RNA, well, we get messenger RNA 1 from one gene, messenger RNA 2 from another gene, and messenger RNA 1 makes protein 1, and messenger RNA, and that protein might do something, like form a structure in a cell, like a part of the skin or part of the cytoskeleton of the cell. It might be do some work. It might be an enzyme that catalyzes some kind of biochemical reaction that's important for life. And of course, there are tens of thousands of these proteins, different proteins, making different parts and doing different kinds of work in the cell. So how do those proteins get deployed specifically in specific cells in a way that enables the animal to make its proper shape and function? Well, there are other proteins that do regulation, okay? They don't make so much structures, but they can interact with a protein to turn that protein on or off or make it do one thing or another thing. They can interact with the mRNA to govern how rapidly a protein is made. They can interact with the DNA to determine whether or not the gene is expressed at all, or at a low level or a high level. So this is a regulatory gene, and the gene that's being regulated is often called an effector gene. And of the 20,000 genes or so in the human genome, thousands of them are regulators of other genes. And in some cases, regulators regulate regulators, as we'll show you an example of that in a little bit. So the way we study them is do experiments. We study mutants. So if there's a regulatory gene that we want to understand, if we make a mutant that changes development in some fashion, we imagine and hypothesize that that gene could be a gene that regulates something, a developmental process of interest. Now, a mutation allows us to find the DNA encoding the gene, and that allows us to figure out what the RNA and protein products of the gene, and then that enables us to connect molecules to developmental processes, okay? So now the mutants we started with, and this is a long story, it begins back in the 1980s or so in Bob Horvitz's lab. We started to study some mutants that had very interesting phenotypes, as we call it. The results of mutating the LIN4 gene is that the animal grows up to be something that looks like an adult and actually has a fertile gonad, but most of the structures of the animal are still juvenile. The animal had gone through 5 larval stages, but at the stages 2, 3, 4, and 5, it repeated the production of structures that normally would occur only in the first stage. And so it's missing all kinds of structures that should be there as an adult. So well, let's call it a persistent juvenile. Now, the opposite phenotype is exhibited by mutations in this other gene, LIN14. And here, this LIN14 is an animal that should be a larva, but it's actually an adult. Because what it did is it skipped early larval development and went straight to later larval and then adult development. So I think you can see that we were super interested in mutants and therefore the genes, because we figured, well, the LIN4 gene and the LIN14 gene might be parts of a developmental clock. And that clock ultimately regulates the production of juvenile genes versus adult genes. So it's regulating gene expression in a way that enables the animal to deploy the right parts of the animal at the right stages and therefore make the animal come together properly. So we began with this hypothesis that we had a regulatory pathway that was a clock-like, had clock-like character. So we wanted to find the products of the LIN4 gene and the LIN14 gene. So the Ruvkin lab started with a LIN14 mutant and by molecular biological approaches identified the messenger RNA and the protein, the LIN14 protein. And we in the Rosalind and the co-worker Rhonda Feinbaum in the lab identified the LIN4 gene and its transcript, and it turned out it had a super unusual transcript. So this is the whole microRNA story, begins with discovering that the product of the LIN4 gene is not a protein, it's just a little RNA, exceedingly small, but it's complementary in base pairing with a part of the LIN14 mRNA. And what that does, when it binds to the LIN14 mRNA, it reduces or extinguishes the production of the protein from that RNA. So the LIN4 messenger, the LIN4 microRNA is a repressor of LIN14. And that's how this little clock works. Now, LIN14 being a regulatory protein, it regulates juvenile genes, and that's how it specifies juvenile traits. So Lin4, by turning off Lin14, enables the animal to transition out of the juvenile stage and onto the adult. So now we know how microRNAs work, and the Lin4 and Lin14 case are an example of how messenger RNAs are produced from genes in the nucleus of the cell. They go out to the cytoplasm where they're translated by ribosomes. And the Lin4, or a microRNA, is produced also from an RNA transcript that gets processed by a series of scissor-like slicer and dicer proteins rather, that produce this small RNA that associates with a protein called arginine. And that protein and its microRNA partner find their target mRNAs through that complementarity. And by binding to the target, it elicits a regulatory response of various kinds, and that's an area of active study, how these various effects of the microRNA and arginine present on the mRNA governs the production of protein from that mRNA. So Lin4 was the first microRNA identified in C. elegans. And there it is. And then Let7 was identified in the Ruvkun lab, also in C. elegans, as a second microRNA, also controlling parts of this developmental clock. And then Ruvkun discovered, in fact, that Let7 was actually present in the genomes of all the bilaterian ancestors, and therefore that microRNA evolved hundreds of millions of years ago. Now, then quickly, many other microRNAs were identified, including Lin4 was found to be in all those genomes. And so therefore, Lin4 was amongst a whole series of microRNAs that seemed to evolve back when we became bilaterian. And there's even microRNAs that are even older, like miR-100. So the discovery of microRNA was an accident, you'll notice, right? Because we were just curious about genes that control developmental timing. We're not looking for microRNAs. We came across the microRNA because it was there. And so, and now we know that microRNAs are also in human genomes, and many of the same microRNAs are in worms and in humans, and that they control human development and physiology, as I'll talk about in a moment. So this is to remind us all once again that curiosity is the fuel of discovery. So microRNA-mediated networks then turn out to be fascinating aspects of gene regulation in animal cells, including humans. Human DNA encodes hundreds of microRNA genes, different ones, and 25,000 protein coding genes, and each microRNA, in many cases, can regulate thousands of protein coding genes. So microRNAs mediate the flow of gene regulatory information in very complex and fascinating ways. And this has pervasive physiological impacts. MicroRNAs have been shown to affect cell proliferation in many, many different contexts. They can regulate cell death of various types. They're involved in the development and maintenance of organs and tissues. They're involved in wound healing and repair. So microRNAs have been implicated in human diseases in lots and lots of contexts. So there are tumor suppressor microRNAs. For example, microRNAs that are aberrantly downregulated or depleted in tumor cells, and that helps promote proliferation of those tumors. There are also oncogenic microRNAs that act in the opposite way from tumor suppressors. Actually, their aberrant expression, often by gene duplication and overexpression in that fashion, can drive tumorigenesis. There's also fascinating applications of microRNAs in neural development. These are identified and for us by human mutations in argonauts, which is, you may recall, is the partner protein for microRNAs. So neural developmental delay can result from a very pervasive disruption of microRNA activity in the developing human. And the final example, though, of human disease applicability of microRNAs, I want to use as an example this microRNA 17 story. And what I want to emphasize here is how much work goes into establishing an understanding of the involvement of, say, a microRNA in a human disease relevant physiological context. Notice all the authors involved. This is in 2016, so this was almost 15 years after this particular microRNA was discovered in humans. And so the paper is this, I'll refer to it as Hudson et al., 2016. And what Hudson et al. Determined that in polycystic kidney disease, which is essentially an overproliferation of certain cells in the kidney, it's a kind of tumor, but it's a fatal condition because it disrupts kidney function. This arises genetically from loss of one gene for the PTK1, because all of us have two PTK1 genes, and normally the normal complement PTK1 genes makes a certain amount of mRNA that makes a certain amount of this protein, polycystin, and that is engaged in a feedback loop with a microRNA and also is an inhibitor or a manager of proliferation in the normal context of kidney repair and regeneration. So now when you have too little of the gene, you have less mRNA, less protein, and in fact, you have more miR-17, and that reinforces this aberrant low amount of polycystin, and this leads to hyperproliferation and the pathological condition. So what Hudson et al. Showed was that in fact miR-17 is an Achilles heel of this disease condition. Because if you were to knock out miR-17 or inhibit it in some fashion, that restores much of the expression of polycystin and corrects the hyperproliferation, right? And so this suggests an ADPK therapy, which would be some sort of inhibitor of miR-17. And what they devised was a small nucleic acid therapeutic of precisely the kind that are developed here in Anastasia and Watts labs in the RTI. And this one is designed to basically mask the microRNA such that it cannot find its targets. And that enables the low polycystin and high proliferation condition to be restored to normal. So Novartis has acquired Regulus, and Regulus is the one who developed this. And the fact that they paid hundreds of millions of dollars for this therapy means that they have confidence that it should work in people to correct this disease. Now, what I want to emphasize here is this is April 30th, 2025, and the paper, remember, was 2016. So now we've got almost another decade between the biological insights, right, in Hudson et al., and the actual realization of the therapy. So it takes a lot of people and a lot of work to make real the viable, let's say, practical applications of a therapy, in this case, for a microRNA. So in my last points I want to make is I want to return to our US scientific ecosystem story here. And let's reflect on the last point I made, which is, it can take a lot of time and there's a lot of uncertainty about developing therapeutics from basic science biological discoveries. 1993 was the microRNA. It was 2000 before miR-17 was discovered, 2016 before the key fundamental insights were really worked out that enabled the designing a therapeutic, and then another 9 years to make the therapeutic, okay? And so our investment, the taxpayers' philanthropic investment, is really that. It's an investment in the sense that it will take time and an uncertain amount of time to realize the benefits of what we do and how we support what we do. So there's also, of course, the economic stimulus that is coming all the time, and that's not a lengthy and uncertain process. That can be less lengthy, but even more immediate is the trainee, let's say, flow through the laboratory. Because these individuals are people who are passionate about science. They're generating new ideas. They're learning how to become, independent investigators, becoming productive and talented and creative members of the scientific workforce. And so, you know, throughout my career, oh yes, economic stimulus. Yeah, so Candy's pointing out that I forgot to say the economic stimulus is part of that is is the fact that looks crass. Really, what comes out of this is, of course, relief from suffering and all the human payoff that comes from that. Because of this uncertainty and the length of time it can take for the payoff of basic research to manifest itself, throughout my career, I have always focused on what I knew to be the immediate impact of what I did. And that is training people in the lab. So, you know, every day we go to the lab and we witness people working their best, learning their best, advancing in incredible ways through progressing as a scientist, an investigator, and a person. And people come out of the lab doing research and they go right on to something else, the next stage. And what we do in the lab enables them to do that. What they did in the lab may take decades to pay off, but what we help them do in the lab has instantaneous payoff in terms of realizing the dreams of young people and contributing to a trained scientific workforce. So I think that, you know, as supporters of basics of science, biomedical science, we have the opportunity to invest in the long term. And we also have the opportunity, let's say through fellowships and scholarships, to invest immediately in people. And the wonderful thing about these people is we see them in the here and now, and we get to know them in the here and now. And that's a wonderful, incredibly rewarding kind of interchange or exchange between those that support the students and postdocs and the students and postdocs themselves. So I hope I didn't go over time. I want to thank you so much for listening and being here with me today. Thanks so much for all that you do for all of us. Yes, stop share. Okay.
Thank you, Victor.
Hi, Thoru.
I think your idea to link the laboratory ecology to a landscape of our profession was brilliant and was very informative, I think, for our lay members in the audience. I just wanted to also say that there's a quality you have that I've seen in other really successful scientists, which is like you stayed in kindergarten. You know, there's an open mind, there's an innocence, but yet there's the capacity to be awestruck.
Mm-hmm.
And I think you display that in your life in a very impactful way. And thank you so much for that. Kim Canty has been monitoring the chat room. And Kim, I would like you to let us know if there's any questions that are there.
We did get a few. How do you balance basic research with the potential for practical translational applications?
Well, this part of what we do is facilitated in a huge way by the institution, by UMass. So, you know, I'm not trained as let's say a therapeutics developer. You know, I'm not a bioengineer. And so I do basic research for the reasons sort of we outlined, because, you know, we're just really fascinated by the processes we're studying. And so what happens is, you know, when we produce a finding, then there are colleagues here at UMass that we can work with, and they actually come to my office, you know, and seek people out, you know, to explore what kind of, you know, practical applications might be, and, you know, might come from the work. And often that can involve, go as far as developing patents and intellectual property that can then be, be enabled, that stuff, the work to be done to actually translate it. Because there's collaborators at UMass who work in this area. So for example, we've been working with that Argonaut syndrome and genetics, and we are thinking about possible therapeutic approaches to address the disease, but that's outside of our expertise. And so we just walk across the street and talk to Anastasia Kovarova, and we have collaborations that are beginning where we are collaborators with, the world's expert in this. So this balancing of basic science and translation is something that is I can't say enough of how that is manifests through the university or the institution setting up mechanisms and bringing collaborators together on the same campus, or potential collaborators who are on the one hand, let's say, therapeutics engineers, and the other basic scientists. It's really all about the institutional culture and ecosystem on the campus.
But including the clinical staff.
Yeah, also
who come to us with ideas about how we can help them and help their patients.
Yeah.
So I mean, we don't know anything about human physiology or medicine, but they, they bring us their ideas too. It's
really a unique institution, I have to say. I mean, it's not an arts and sciences campus; it's a medical campus, and there's an enormous strength in basic science. I mean, I think there's 13 C. elegans laboratories on campus, and that's more than any institution anywhere in the world. Okay, that's a medical campus, right? And it works, for all the reasons Candy mentioned and that I was saying.
That's terrific. What are the biggest challenges in understanding the precise role of microRNAs in gene regulation?
The biggest challenges is that these microRNAs are way more versatile, way more nuanced than we could imagine. And they are engaged in so many biological processes in the cell that, I try to give you the sense of how it's all about networking. Like there's so many hundreds of microRNA genes, and each one regulates hundreds or thousands of other genes. So this is a network. And each node of the network is connected to and touching many, many molecules. And so it's what we're discovering as we go along is that we're constantly surprised by the amazing things that microRNAs can do, and the amazing ways that they will be engaged or connected into particular biological processes that might interest me or somebody down the hall or somebody else. So almost no matter what sort of physiological angle an investigator takes on biomedical science research, microRNAs will be involved in that process. So that's actually a wonderful aspect of studying a class of molecule that touches so many things in the cell, because everybody's sort of interested in the same thing.
If you could go back and advise your younger self at the start of your career, what would you say?
Oh my God. Yeah. Oh my God. So Candy can tell me plenty of things, but I mean, it would be like, wake up, man. You're such an idiot, I mean, I wouldn't change anything, frankly, because here I am, and I really can't. I can't. Yeah. I can't complain. So I mean, I think that I would, yeah. You know what I mean? I wouldn't change any of my decisions. I would just tell myself to be a better person,
That's good advice for everyone, right?
Yeah, right.
Have your lives changed at all since receiving the Nobel Prize?
Yeah, it's changed a lot. Yeah. That was a big surprise, and so things changed sort of instantly. And what was most one of the most striking things, we arrived at campus and there was a camera team walking backward across Plantation Street as we walked to the lab, you know, and then people were, you know, people were just so overjoyed, and everybody we met at UMass, you know, they were just so happy, and so I suddenly realized that, hey, this isn't about me. It just makes people happy that this happens, you know, and so that made everything, you know, then I could relax and really, you know, enjoy it, because, you know, I wasn't as self-conscious as I might have imagined I'd be, you know, so, yeah, and what else? You know, we just met amazing people, you know, we got to meet actual Nobel Prize winners, Kim, you know,
Yeah.
we got to be in the same room with them. It's awesome.
I think the room might have a question, but I just have one more here. What
sure.
continues to drive your curiosity?
Well, yeah,
It's never ending.
yeah, I mean, it's impossible. I mean, because, you know, in our field and everybody's field, you know, everybody we hear talk, we go to talks at, you know, at UMass, you know, every day, a student or a postdoc or another faculty, whatever, and they're talking about stuff that is just amazing biology, you know, and it's new and it's surprising and it's intriguing, you know, so it's irresistible. You know, I just find it irresistible, and you know, I think, you know, curiosity is something that's it's human nature. You know, we've all seen it in you know, in toddlers, right, in children, right. It's what drives them to learn about the world, and, you know, so it is natural, but it can be inhibited, right, and it can be nourished, you know. And I think that I've been lucky to have curiosity nourished, you know, first by my parents, and then by going to, you know, by teachers, and, you know, and going to MIT where everything's science and everybody loves science. It just is, you know, it's fantastic. So, and then ever since then, I've been around scientists and I love scientists. So it's, your curiosity is always sort of reinforced and nourished by experiences.
It's clear that you and Candy have an incredible relationship that's pretty unique to work so closely together. How is that for both of you? Do you talk about work all the time? How does that?
Well, Candy, how is it?
Yeah, there's you know, been ups and downs. But, you know, it's much more, you know, since coming to UMass,
Easier. Yeah.
it's far easier, you know, so there's just a lot more support from the administration, from UMass, for, you know, the little things like, you know, the air conditioning working, you know, and stuff like that, which I had to deal with in other places. And also you just don't, you feel like, you know, this is your family, and the people, you know, like Victor said, like, people were happy for us.
Mhm.
Right. And, you know, there wasn't this kind of vibe you can get on other campuses where people are like, you know, just not, you know, okay, Victor had this prize, and, you know, that, well, that's him, you know, it has nothing to do with me. Right.
Yeah, that's what
People were
surprises
just
so much. Yeah.
happy, you know, and they felt it and they owned it too.
Yeah.
And that's sort of what we feel.
Strangers owning this prize.
Yeah.
That's
Yeah.
what was astonishing and quite, you know, very interesting. It means that everybody has sort of a hunger to know of something special, or to see something special happen, you know, and in particular to see science celebrated, you know.
Yeah.
There's a great hunger for that. I think there's a hunger, you know, that's at the university, on a campus, but I think there's a hunger out there in the public that just is they don't even know that they want it. You know, they want to understand science and celebrate science, but they don't know that they want it because they just don't know. And we haven't talked, you know, we haven't communicated enough about how important it is. And I mean, you notice that the Massachusetts taxpayers and the American taxpayers, they are fueling this, right? And I just wish that they knew how important what they do is, and how and just take the justifiable pride in it. I think we have most of the populace just unaware of something that they should justifiably be very proud of.
Mhm. I think, Dean Flott, do you have a question?
Yeah, I believe Professor Peterson had his hand up first.
Oh, okay.
I want to defer to my colleague.
Okay. Yeah, so a couple of unmuted. Sorry. Yeah, so about candy. I think one of the reasons that Victor and Candy came here was because there were many of us in the community of RNA science here who recognized her role in the great discovery
Mm-hmm.
that both of them made. This was not obvious to many other institutions, but those of us who work on RNA knew, yes, that was that's how it happened.
Mm-hmm.
Victor was kind to mention the student, the very brilliant student who was also involved. I also want to just quickly say that I had the honor of introducing Victor at a lecture at the Marine Biological Laboratory in Woods Hole, probably 7 years ago. And I mentioned something in the introduction about a company that UMass had formed around RNA. The company calls itself Alnylam. And Victor got up to the podium and he said, Thoru, thank you for remembering that. I know that constellation. Turns out this is a polymath guy. He knows astronomy. He mentioned it in his childhood. And he said, I know that constellation up there in the sky. I thought to myself, this guy is not just about RNA. He knows a lot.
That's why there's one other fact that I know. Yeah.
Last thing I want to say is that Victor talked about his childhood in Vermont. He did not talk about his parents and grandparents in Eastern Europe. And I don't want to ask him to go
Yeah, well,
into.
my dad was a Polish immigrant, right? And
I think
he was a refugee, and so as a child, we were very aware that here was this talented and brilliant man whose education was terminated by the war, in the 10th grade, right? And then he heroically kind of made his way to the States eventually. Yeah, so we were always aware that we were privileged, like, oh man, he had it so tough that we better do well, I was actually obsessed with trying to impress my dad throughout my childhood. So
I didn't
yeah.
want to necessarily evoke any personal memories, but just to say we are a country of immigrants.
Oh yeah.
God bless us.
Now Candy's parents are immigrants from China.
So let us,
yeah.
I don't want to make a benediction here, but let us remember that.
Yeah, well, I have another slide I could have shown you, which was the lab at its peak, where there were 12 people in the lab and they were from 8 countries, so
there you go.
that was the peak into
International.
lab.
So, but Thoru, you only knew about my contribution through a sort of sneaky way, because Craig Mello, the other Nobel Prize winner at UMass, was our graduate student.
Oh yeah.
And that used to be the point of pride for us,
right?
That we could say, it's like when your kid produces, and stars and shines. And so, we were already proud parents. And,
exactly. Yes.
Yeah, so Craig was in our lab when the discovery.
Nomenclature is very complicated because what, in this case, with regard to you and Victor and Craig, are the P0 and the F1 and the F0,
Yeah, right.
all of the above.
Well, yeah, and then Craig, recruited us here. So
Right,
what does that
right.
make?
Yeah.
So, yeah.
Kim, I didn't mean to preempt any other questions. So what's in the chat box? Anything?
I think Dean Flott has a question.
Yeah,
Oh,
well, I just wanted to, I had to chime in about the jubilation you felt on the campus about your announcement of the Nobel Prize, and really for both of you. It is very much spontaneous and very much an expression of both of pride in the institution, but I think also of people who know you, who know you both and know how wonderful colleagues that you guys are. And so I have to thank you for making the decision to join us 18 years ago in 2007, and for I would be thanking you even if you hadn't won the prize, because you've been such a wonderful colleague to so many of our faculty. Everyone on campus knows that. So good that our Hudson Hoagland Society members know that. But I also think just one last thing, it's that we're a teaching institution, and the inspiration you give to the graduate students it really is really quite remarkable. Not just because you win the prize, you won the prize, but because when you got an unexpected result, you didn't choose to ignore it because it didn't reinforce what you were looking for. You didn't choose to say the experiment didn't work because you couldn't find a protein. You chose to believe and trust in your data, and that really inspires the students to do the same. So I think that's really one of the wonderful things about having faculty like you in close proximity to our students, because they not only learn that, but they also pick up this vibe of taking joy in the success of all of our colleagues. So thank you for that, and a wonderful influence you're having on our young people.
Well, thank you for those words, but right back at you. I'm really super happy to be here, and super happy to have come. So. Kim, are we done with superlatives?
Yes, we have no more questions. Thank you.
All right.
Thank you, Dr. Flott, for those thoughtful comments. Thank you, Chair Collins. Thank you, Victor and Candy, for being you. That's the best way I can express my heartfelt feelings.
All right. Well, thank you.
Thank
That's
you.
easy. Thank
Hudson Hoagland Society donors
Okay.
for being with us tonight for this very special annual meeting. Thank you all, and be in good health.
Yes,
Yes.
everybody
Yeah.
stay safe.
Good night, everyone. Thank you, Kim.